It's my deep pleasure and my honor to introduce Professor Maurizio Corbetta, who is giving today the keynote speech. Professor Corbetta is a neurologist, and I would not cite all the steps of his career because better he speaks about science and I speak about some educational steps. But I should say I'm quite impressed because he is one of not so many people in the world who is combining clinical work and research in patients and healthy subjects, but he has also a very strong bioengineering background. And this was also a reason why he was capable to develop very important methods, and we will hear perhaps today about the dark matter of the brain. We will hear about resting state connectivity, where he was really a pioneering person and pushed this forward. And now he is collecting and organizing a collaboration across whole Europe where unique data sets are being connected across different hospitals. And those of you who have worked with patient data and who know how special hospitals can be when it comes to sharing data and collaborating, they know that this is really a big effort, but it's also important to join forces in this European network, to better understand what are really the deep characteristics of a certain disease, what are the pattern mechanisms, and then based on that also develop methods that are uniform, that follow a certain protocol in different countries. So there are many merits, and at least some of them we will understand in a deeper way today. So thank you very much for being here, also for hosting us. Professor Corbetta, the floor is yours.
Thank you, Catherine. Thank you so much. Thank you for the invitation. And as I said, it's really a pleasure having you here in Padova. And I hope you have enjoyed the city, which I think is really quite a special city. Okay, so if we can have the first slide, which is the title slide. So today I'd like to tell you about what I think is one of the most fascinating topics in neuroscience today, which is what is your brain doing when it's not doing anything? And what is the inner life of your brain? And that's kind of a mystery right now, but there is a lot of interesting data coming, and hopefully we will have a theory. And I think this theory is not going to be only important for the static organization, but I think it's going to be important for understanding behavior and cognition. So that's what I will try to tell you. So let's see if I can get first my slides. Okay. So first of all, spontaneous activity, and I will define it here in the next slide, is not a property just of the human brain. There is spontaneous activity across the evolutionary tree, from C. elegans all the way to humans. And so it is a fundamental property of the brain. And I can tell you that if you take a slice of hippocampus and you put it in a dish, and now you implant it with 1,000 electrodes, that slice of hippocampus, or even neurons in isolation, will produce spontaneous activity. So the tissue, the nervous system, is the nervous tissue, is basically an intrinsically excitable tissue, which will produce spatiotemporal pattern of activity in a slice that looks a lot as the entire brain. So there is actually something about what is called the fractal properties of the brain, where properties that you see at the slice level, you can capture them at the whole brain level. And of course, between a slice and a brain or a nervous system, the difference, of course, the fact that you have connectivity that is organized in that activity during development, during their prenatal period, during development, and then during individual experience. And so given we are in the Western world, we think we're all different, we're all special. But if you're in the East, of course, we're all similar, right? And I think my take on this is that I'm more interested in commonalities than differences, and I think we're more similar than different. And I'm going to show you some fundamental common properties of spontaneous activity. Okay, so first of all, we do need some definitions, and because definitions are important. When you read papers, you heard, you can read about spontaneous, intrinsic, endogenous. These are all, in my view, synonymous, okay? And it's basically, from an operational standpoint, it's basically activity that is recorded in the absence of a stimulus, in the absence of a task, when the organism is awake, but could also be asleep, and it could also be under anesthesia. Of course, these states are behaviorally different. They're modulated in a different way, and there are fundamental differences when a brain is awake versus a brain is asleep or under anesthesia. But the point is, it is intrinsic. And in the early days, when we start studying spontaneous activity, there was a lot of debate. Well, this state doesn't really exist. It's just basically poorly controlled cognitive activity. But I will show you that that is not the case. Now, we know, of course, as I said, you take a piece of tissue, you put it in a dish, you have spontaneous activity, and we know that organism, newly formed nervous system, will develop spontaneous activity intrinsically, and this intrinsic activity is fundamental for maturation. It's important for synaptic, for axonal guidance, is important for synaptic development, and is also important for myelination and the formation of networks, okay? So there is a lot of activity going on even before you ever experience any visual information, sensory stimulus, or motor stimulus. In fact, there is some beautiful work in prenatal pups, mice, in which you can see, for instance, that in visual cortex, let's see if this starts. In visual cortex, you have spontaneous activity recorded with calcium imaging in prenatally, okay? And the spatial pattern of that activity actually resembles already before the pups open his eyes, 10 days before birth, okay? Basically already resemble the organization of orientation columns in the awake adult animal. So, and this is a fundamental point. The spontaneous activity is kind of a precursor, some kind of a scaffold of what might be happening during the awake state, during cognitive activity, okay? Okay, now, but I'd like to talk about adult brains, and I'd like to talk about human imaging, because I think human imaging has been instrumental over the last 30 years in basically showing us some principles of organization of spontaneous activity. And so this is now a pretty well-known movie that was generated by Justin Vincent at WashU around 2008, and it's basically showing a subject in an fMRI scanner is being recorded at rest, eyes open or eyes closed, doesn't matter, could also be fixating. And those patches of activity, which early days were thought to be hemodynamic artifacts or physiological artifacts, reflect the size of those patches are about the size of human areas, okay? And when you see orange, it means that that piece of cortex has a positive correlation in its Ball signal with the primary motor cortex, which is located about there. And when you see blue, means that there is a negative correlation with the primary motor cortex. And so immediately you see here something that is very different from the picture of the brain that we have from studying the brain when we're stimulating or when we're moving, which is about 150 years of neuroscience, which basically the primary motor cortex is communicating with the whole brain. There is not an encapsulated motor system, okay? Yes, there are supplementary motor area and premotor regions, but basically the primary motor cortex dynamically is connecting with the rest of the brain. And I think that's a very important insight. So what I'm going to show you next is and so that's the primary motor cortex. So I'm going to show you 3 lines of evidence indicating the functional importance of spontaneous brain activity based on the literature of the last 25 years, basically. And I'd like you to share with me the wonder that I think we experienced as we were studying these brains at rest, and some of the insight which are really quite remarkable and quite counterintuitive with the way we were thinking about what is a brain when it's active, what is a brain when it's at rest. And so the first insight is about metabolism. The second insight is about the spatial and the temporal organization of spontaneous activity, and the rest and the similarity between task and rest patterns. And then the third, which is also very important, is that surprisingly, spontaneous activity in the absence of behavior strongly correlates with the way you do things, okay, or the way you are, your individual cognitive variability. And that's very important because when we test behavior, we're moving, but yet when we're not moving, your brain is already anticipating what you might be doing. So let's start with metabolism. This is one of my favorite slides in neuroscience, and it's basically showing a PET MR system where we can get structure and we can get function, and function in this case is fluor-18 fluorodeoxyglucose. And you see when you measure metabolism, how the glucose distributes across the brain, across the body. And what is incredible is how the brain is capturing all this glucose as compared to other parts of the body that are much more represented in terms of surface area and volume. Take muscles or take the intestines, okay? So 1 molecule out of 4 in the adult brain goes to the brain. 1 molecule of glucose out of 4 goes to the brain. The brain is only at 2% of the body weight. And of course, you see also there is a little activity in the heart, and there is a little activity down here. Nothing exciting. This is the FDG draining from the bladder, okay? So basically that's, now if we quantify this is a nice paper in Current Opinion Neurobiology. This is the body mass of the different organs, and this is the consumption of energy across the organs. And you see that the brain is using 21% against a 1.9% of body weight. So where all this energy is going, okay? And David Otwell is a physicist who has been doing some really cool calculation of energy consumption, starting from how many moles of ATP you need to generate a spike. And we know, of course, from many evidence from, even from Sokolov and other people, that a lot of the metabolism is actually at the level of the synapses. It's not at the level of the body, it's at the level of the synapses in this interesting neurovascular unit that include astrocytes, neurons, and blood vessels. And when you go and do this theoretical calculation on the cerebral cortex, it turns out that about 25 to 40% of your energy goes in keeping what they call housekeeping operations, changing the synapses, changing the mitochondria, changing the microtubules, protein production, etc. About 60%, 50 to 60% is in spontaneous activity, which is either maintaining resting potentials or subthreshold potentials, and only 10 to 15% is spikes. And the reason spikes occupy so little of the energy budget is that if all the brain was spiking, the amount of energy necessary to keep it would be enormous. Spikes are metabolically very expensive, and so nature has found the solution to have sparse spike encoding of information, maintaining very high level of subthreshold activity. And if you go into the cerebellum, you find exactly the same thing, the same distribution of metabolism across these signals. So the point that I want to make is that resting potential and subthreshold potential are occupying between 50 to 70% of the activity. So most energy is spent on resting and subthreshold activity, not spike, is being computed, calculated, estimated that about 1% to 10% of all neurons are active at any given time. Now, the cerebellum is particularly interesting as a structure, as we heard also from Alan the other day, because there are a lot of neurons, and when you go and do this calculation in the cerebellum, you find something very interesting. You know that the cerebellum is important for motor control, and the motor control is exerted through the Purkinje cells. The Purkinje cells are the output of the cerebellum that is modulating the thalamus and the motor cortex. But the Purkinje cells are occupying only 18% of the energy, while the majority of the energy is actually coding potential sensory motor associations, not the actual movement, but is actually coding states between the cortex and the cerebellum, between the spinal cord and the cerebellum, of possible combination of sensory motor patterns. And I think this is a fundamental insight that comes from these calculations about what the spontaneous activity is doing. So I want you to make a note, coding of potential states in the brain, not what you're doing, but the universe of the repertoire of potential sensory, motor, and cognitive states. This idea that the spontaneous activity occupies a lot of the energy was apparent in the early days of PET activation, oxygen 15 activation studies. And we kind of ignored that for 20 years. Why? Because if you take a subject, you put him in an MRI scanner, and you flash the subject visual system with an incredible unnatural stimulus, a flashing checkerboard that is flickering at about 10 hertz, which is basically flicker fusion. After at 12, 15 hertz, you don't see the stimulus flickering anymore. At 8 hertz, 10 hertz, you still see it flickering, so your visual system can keep track of that, but it's very unnatural. We get changes in visual cortex between a stimulation condition and a control condition of about 40% changes in blood flow. But if you use a natural stimulus, like saying a word, the changes in the frontal cortex are about 1%. And if you take the difference between seeing something and not seeing something, a task of consciousness, there is no change in metabolism. And of course, when Mark Rakel and Peter Fox and they start doing these activation studies, everybody was puzzled by this, because these two brains, they look very similar. And if you didn't know this was a stimulation and this was a control, you would be hard pressed to see where was that visual activation. And so it came this idea to do, as in EEG, to do event averaging. And event averaging suppresses out all the spontaneous activity, and it leaves you these tiny task-related signals that trial after trial, you're averaging to look at these activation spots. And so we did activation mapping for 25 years, basically looking at these tiny signals, and everybody is his own area. I have the dorsal and the ventral attention system, and other people at the memory system. Everybody name all the brain with all these names based on these very tiny task activation signals. But we completely ignore what's going on underneath, the baseline. So Mark, which is smart, super smart guy, is kind of start thinking about this as well. But this is like he made this analogy with the dark energy of the universe, the dark energy of the brain. Like dark matter, you don't see most of the universe matter, and it's dark, and the same is in the brain. Now we know, and so I think a very important question in science right now is where this energy going? And in my view, I think a very strong goal, which I hope we can get done through eBrains, is for every voxel in the brain, for every brain region, every vertex, that we can write an equation where you have a contribution from structure, a contribution from function, a contribution from power and electricity, and synchrony, and try to explain the metabolic expense of that particular brain region. And I think this would be a very strong goal that we might be able to achieve, but there is already evidence, for instance, in Drosophila, that if you take a Drosophila brain and you can measure basically all the neurons in Drosophila, so there is no point here that you're missing something. And now you have three kinds of reporter. You have metabolic reporters of aerobic metabolism, ATP, anaerobic metabolism, pyruvate, and then you have activity through calcium imaging with a resolution of about 30 hertz. And you see these correlation matrices between activity and metabolism. So this is GCAMP versus ATP, and this is pyruvate versus GCAMP. And you see this beautiful correlation between the level of activity, the connectivity measured with calcium, and the level of metabolism in these neurons. And what is interesting is this correlation occurs only for activity below 0.1 hertz. If you go above 0.1 hertz, there is no correlation. So this is very important. The correlation with metabolism occurs only for the ultra-slow activity in the brain. And what is particularly interesting in this Nature paper is that when now we are electrical stimulating some of these neurons are firing off fast neural activity, but then over minutes, that circuit will start accumulating ATP in anticipation of potential future use. And this is also very important inside, that the metabolism might be a prior for future activity. And so that our brain is set up in a way to optimize future energy consumption, and the future energy consumption may be coded through the ultra-slow activity of the brain. Now, you can do this kind of work in human imaging, and so we're starting to do this in Padua. When I arrived in 2017, we set up this, what we call a brain map protocol, in which we simultaneously collected FDG and BOLD to try to explain the relationship on a PET-MR hybrid system, to try to explain the relationship between the two. So here's a paper that was published by Tommaso Volpi, who was a PhD student with Alessandro and me, in Journal Cerebral Blood Flow and Metabolism a couple of years ago, in which basically we got fMRI data that were simultaneously acquired with PET data, and we extracted something like 70 variables, static and dynamic variables of BOLD signal. BOLD peak amplitude, RIO, ALF, BOLD variability, static functional connectivity, dynamic functional connectivity, eigencentrality, between centrality, whatever you can think about, how can you measure the BOLD signal, and also measure of SUVR across multiple cortical regions in subjects. We had two data sets, so we replicated this data in two independent cohorts, and we had basically different models to try to relate the two sets of variables. And so this is the correlation of the BOLD signals, the BOLD variables, and you see that all the signal BOLD, local BOLD signal variables are strongly correlated, for instance, with the static functional connectivity, and they're negatively correlated with all the dynamic functional connectivity measure. So this is a very interesting, to some extent expected. The more you have static functional connectivity, the more those areas are related to local signal, but the less are related to switching signals. But down here, you see the positive correlation with the SUVR. So the SUVR seems to be strongly related to local activity and static connectivity, and inversely correlated with areas of the brain that are dynamically switching. And I think that makes sense, because if you are averaging activity over time, those connections that would be more constantly active, they will consume more glucose. If you keep switching, you're active only part of the time, and so you're consuming less glucose. And when you do now model selection, and you try to relate these two sets of signal across very complicated model selection that Alessandra Bertoldo and Tommaso developed, you see that the signal, the RIO, which is the local synchronization at the voxel level, is explaining a lot of the SUVR-BOLD relationship. You also have other mild contribution of other features like static between centrality, but also you see negative correlation with indices of variability in the local response, is also in the connectivity, in the global connectivity, like the variability of the between centralities, which is an index of dynamic connectivity between networks. So this is a first approximation, but imagine that now you can get a data set in which you can measure structure, you can measure myelin, you can measure glucose, you can measure fMRI, and you can measure EEG simultaneously with high density, and this is exactly the goal of the 5M, the pentamodal connectome that we're going to try to do through eBrain. So we're going to do 200 subjects across 5 centers to basically try to get to this issue, where is the energy going? All right, so interim summary up to this point. I hope I show you that the high metabolic consumption of brain tissue comes not from spikes, but from spontaneous neural activity and connectivity at low frequency, less than 0.1 hertz. And I think this is the first kind of take-home message that I'd like you to have. Now let's talk about spatial and temporal organization. Now this is much more known because this has to do with brain networks, resting state network, blah, blah, blah. I'm going to just show you, however, some interesting insight that we had over the years. So first of all, we have to start with the traditional neurophysiological model. In the traditional neurophysiological model, and this is what I'm talking about, Mike Shadlin and Terry Sinowski, about 1980. The normal physiological model is that spontaneous activity is noise, is Poisson noise, and Poisson noise is very important for forward transmission of information. So if I'm doing a sensory motor transformation from the visual system to the hand, Of course, information will activate, photons will activate the retina, electrochemical transformation in the visual cortex, and now I'm going to have this chain of forward events driven by the photons, and these forward events will change, will go more prominently forward, if you wish, depending on the level of noise in the system. So if the postsynaptic neurons in a situation of excitability, it will fire more. If it's in a situation of non-excitability, the noise level is low, it will fire less. So noise is important basically to push forward this sensory motor processing. And this was basically the story until 1995. And remember then from 1988, Peterson et al. Until 1995, for about that many years, we basically did activation studies. Now, Bharat Biswal made this key observation. He's a physicist. Many of you know him personally. He was tuning the scanner in Wisconsin, in medicine. And he was tuning the scanner. He was using what physicists do when they're tuning the scanner. They're asking subjects to move their fingers in the scanner, or they're just measuring noise in the scanner. And so he put up the map of finger tapping task versus control, and then he put an ROI on the motor cortex just to measure noise in the absence of movement, and he measured the temporal correlation of the ball signal with the rest of the brain, and as magic, you see now that the contralateral motor cortex and also the supplementary motor area and the premotor area, all these regions in the absence of movement, they were showing positive correlation. This paper that was eventually published got a lot of problems in getting reviewed, and this signal was basically forgotten for about 10 years, and it was interpreted basically noise and artifacts. But this is the strength of the modulation. So this is a ball signal over time from the left motor cortex and the right motor cortex, and you see how these two signals, in the absence of any movement, and even in the absence of any EMG activity, those two signals are strongly correlated. The correlation goes in the thalamus, the correlation goes in the pons, the correlation goes in the cerebellum. But remember what I showed you with the first movie, that this is a static average correlation in the motor system because the motor system talks to everybody. So yes, over time, we have a motor resting state network, but dynamically, we do not have a motor resting state network. Dynamically, we have a motor system that is interacting with the rest of the brain. So another surprise came from Mike Fox's work, 2005, 10 years after Biswal observation. And we had done experiments on the memory system. And so this is activity when subject, task-related activity, when subjects were actually performing an episodic memory task. They were remembering scenes and words. And you get these maps that you now all know as the default mode network during activation. And then, of course, you then measure activity in the resting state, and you see now that the episodic memory system, what we call now the default mode network, is showing correlated activity. Now we're getting used to this, but this is really important. Your memory system, your hippocampus, your retrosplenial cortex, your medial prefrontal cortex, you're not remembering anything actively, and yet it's synchronized. You go into the attention system. We had done work for 10 years on systems of attention. And now here's a beautiful kind of very single subject map of a subject orienting attention to a particular point in space. This is time-resolved event-related fMRI with a resolution of about 2 seconds. This is a map of the endogenous orienting of attention. There is no stimulus, it's all endogenous. In the resting state, these areas are showing a high level of correlated activity. And we can go on and on and on and on. And you can do what the Human Connectome project has done, which is to ask, okay, how many regions we have, how many patterns of correlated activity we have. And so this is beautiful work, as you all know, by David Van Essen and Matt Glasser, Nature 2016, in which they map out the whole cerebral cortex using this signal. And what is incredible for me, because this is all data-driven work, that it turns out that basically the whole brain can be organized in 5 axes, okay? We have visual areas, we have sensory motor regions, we have auditory/language regions, and then we have these areas in gray, dark gray, and light gray, which have to do with the control of the external worlds, region involved in perception, decision-making, visual-spatial attention, premotor planning, and regions involved in internal control, memory, emotion, body schema, self-reference, theory of mind. Why the brain is organized that way? Why it falls off in this? So that's why with Alan yesterday and with Mike, we were discussing, well, are there some genetic profiling of this? I mean, are there transcriptomics that are related specifically to this major axis of organization? Because it's not necessarily a given that the brain should be organized that way. Now, of course, this organization is not unique to humans. In fact, the mouse have the same, basically, two axes of organization. The macaque has the same two axes of organization. And even in the zebrafish, when you have neurons organized by connectivity, they tend to clusters, not because of statistical reasons, they tend to clusters in discrete modules. Now, the zebrafish, we don't know what is the internal and the external, and we don't even know it in the mouse, but the default mode network of the mouse looks a lot like the default mode network of the human. So it is actually an axis of organization which is probably across species. Now, this is the spatial organization. Now, what about the temporal organization? Now, we know the BOLD as a slow filter, okay, so the activity is slow. And so we keep saying, well, BOLD is slow, neural activity is fast. We cannot really relate the two. But if you look at calcium imaging, now 30 Hertz resolution, you see that the fluctuation across the whole brain show basically the same fluctuation of activity that you see in BOLD, 0.1 Hertz. If you go to EEG, of course, we have alpha and delta and beta and gamma, but you also see this very prominent fluctuation, broadband fluctuation, at 2 or 3 every 30 seconds. And now if you get 10,000 electrodes, neuropixels, inside the mouse brain, you get exactly the same fluctuation. 1 minute, you see 3 or 4 fluctuations, which are this broad swath of activity across the mouse cortex, across the whole brain, visual cortex, motor cortex, cognitive cortex. So this is a very prominent effect, a very prominent signal. Now, how this slow burst of spontaneous activity relate to the task evoke activity? Are they related or not, right? I mean, that's an important question. Or is the task-driven activity somehow a specific example or other things? So this has been studied very well in calcium imaging and voltage-gated signal by people like Tim Murphy, which have given to mice sensory electrical stimulation in the somatosensory cortex. I'm going to go back and restart this. And you see this in the somatosensory cortex with electrical stimulation, which is very unnatural stimulus. You see this very quick burst of activity, 20 milliseconds of activity in the somatosensory cortex. But now, if the mouse is at rest, you see basically that in the somatosensory cortex with a much slower temporal resolution, you see similar events, they're replicating exactly the timing and the distribution, the topography. So here's, minus 200 milliseconds before the events. And if we just let it go, here's the event. And the event is associated topographically similar to what the task is doing. Now, they're not the same. And so when you make movies, dynamic movies of the evoke activity and dynamic movie of the resting state, you basically say get the same topography. So this is not the same because the evoke activity, first of all, has a much higher speed of propagation across cortex as compared to spontaneous. And the other thing that the spontaneous activity is much more variable than the evoke activity. And I think this is very important. The evoke activity becomes stereotypical as compared to the spontaneous activity. So the spontaneous activity represents more states, and when you're doing a task, now you're locking yourself in a particular sensory motor state, much more restricted in terms of dynamics. Now, how many motifs in the brain we have of spontaneous pattern? And this has also been studied in mouse, where basically they've been doing this beautiful experiment in which they're comparing systematically spontaneous activity and task evoke activity for vision, audition, somatosensory stimuli, and even social behavior between mice being housed in the same cage. And it turns out that about something in the order of 50 motifs of spontaneous activity are recapitulating all the possible task-driven states that you can see in a mouse, including social behavior. So I think this is also an important insight that the spontaneous activity spatio-temporal patterns are somehow fingerprints of how your brain is going to activate during the task. Now, if we move up to the systems level, okay, and we look at this at the level of networks, we see exactly the same thing. So here's a single subject. This is a community embedding, is a modular way of looking at resting state activity with all the networks associated with it. So this is a single subject at rest, and this is the same subject when he's looking at a movie, a natural vision movie, and you see basically that these resting state networks are getting reorganized, okay, in space to basically represent that movie. So again, cortical motifs of spontaneous activity are getting reorganized to do tasks. So, in summary, spontaneous activity is organized in a discrete number of spatio-temporal motifs traveling at about 0.1 hertz that recapitulate sensory, motor, and behavioral evoked patterns. Okay, and so that's the second take-home message. The first take-home message is the metabolism at 0.1 hertz. The second take-home message is that task-related activity travels also at 0.1 hertz. Let's talk about behavior now. So I told you that we have this axis of organization, the external world and the internal world. Now, do they have any relationship to behavior? In the Human Connectome project, Smith et al. 2005 performed this analysis in which they got this 200 measure of behavior, including crystallized intelligence, fluid intelligence, task switching, whether you're fat or thin, whether you're healthy or not, whether you're rich or poor, whether you're smoking marijuana or not. I mean, everything that you can think of, as you know, from the Human Connectome project. And now you do a massive principal component, and it turns out there is one axis, which is this axis here, that is basically including all these variables and is accounting for 15% of the variability across 1,200 subjects. So subjects that in our society have higher crystallized intelligence, higher fluid intelligence, higher G, they tend to earn more, they are more educated, they're thinner, okay, and so forth. They tend to have more segregated network between the network of the external world and the networks of the internal world. So in a static analysis, if you have a good segregation of your default network with your dorsal attention network, that through a canonical correlation that of course is maximizing the variance between the behavior and the connectivity is accounting for this. So this is now weights on this particular segregation measuring networks and basically the weights on this particular axis of behavior that includes all these behaviors. So that is incredible. At the global level, the static connectivity is explaining something common across many subjects that is capturing something about our behavior. Of course, I'm sure there's going to be some genotype influence to this. Now, what about circuits? It turns out that spontaneous activity is also capturing learning at the circuit level. So here's a paper that we did many years ago in which we basically asked subjects to change their periphery, 5 degrees of their periphery field, into their fovea. So this task, discriminating L from T's, requires the fovea. If you train a subject 7,000 hours, then that subject at 5 degrees can get 90% accuracy. And if you do that, and you look at the circuit that is activated, you get retinotopic visual cortex, you get the dorsal attention network, etc., etc. And the connectivity in the resting state is getting changed in the task-relevant circuits at rest, in the subsequent rest, after you're performing these tasks. Not only, and I think this is the key point about prediction, but also the way your brain is set up before you do the task is predictive of your future performance. So if you have a good integration in the visual cortices, in the retinotopic visual areas, then you're more likely to reach criterion more quickly than a subject has less integration in your visual system. So this activity at the circuit level is also the way it's set up, is also predictive of how you're going to perform a future task. Okay, the final thing about behavior that I like to mention, there are many possible things that I can mention, is the fact that when you have a lesion in the brain, you have neurological deficits. The neurological deficits are expressing deficits that you see when people are moving, like hemiparesis, or inability to move your eyes, inability to speak. But when you look in the spontaneous activity, you see patterns of abnormal connectivity that is strongly correlated with behavior. In fact, certain patterns of connectivity, like in this case, you have normal subjects, we have normal connectivity between the two sides of the brain, supported by the corpus callosum. When you have a lesion, you have this dyssynchrony in the brain, and this dyssynchrony in the brain at the acute stage accounts for behavioral variability. Again, this is not trivial. The brain, the way your brain is set up at rest after a lesion, is impeding those regions to basically drive activity normally in order to produce normal behavior. And of course, these effects are not limited only to the region that is damaged, but they're actually diffused across the brain. So here's a healthy subject dynamic functional connectivity map. This is an acute stroke patient at 1 week. This is a large lesion in the left temporal lobe, but something between 50 to 60% of your brain connections are actually desynchronized, are not synchronized properly after an acute lesion. And of course, we're using this signal now. This is a grant that we have from the European community to use this signal to map the neurophysiological underpinning of these connectivity abnormalities. And through computational modeling that I don't have the time to discuss, we're going to try to develop new intervention for correcting this abnormal connectivity pattern. And yesterday you heard that these patterns, of course, they're not unique to stroke, but they're basically most of the pathologies, degenerative pathologies, glioma, and inflammation, etc., can be thought of as a connectomics disorder. So the third interim summary of this kind of travel through the past is that spontaneous activity patterns predict global and circuit-specific behavior and their abnormalities, and their relationship with behavior strongly suggests the causal role of spontaneous activity in modulating task-evoke activity. So now, in the second part, so these are the key points that I kind of try to illustrate. Now, spontaneous activity consumes the majority of the brain's energy. Spontaneous activity is organized in a finite set of dynamic spatiotemporal patterns that recapitulate sensory, motor, and behavior evoke activity. And finally, the spontaneous activity pattern predict normal and pathological behavior. So the question is, what is the function then of this spontaneous activity? Why is it there? I mean, how is it serves cognition? And there are theories about this, okay, that have been proposed over the years. The first theory is that spontaneous activity is basically postsynaptic noise that can be organized by the connectivity. And this has been shown in the most, I think, elegant way by these whole brain models, where you take the virtual brain in kind of e-brains parlance, but whole brain models, in which you take basically brain, you derive their structural connectivity, okay, you attach computational units to the structural connectivity, can be spikes, can be local field potential, can be oscillators, it doesn't matter. And you convert that activity through an hemodynamic filter, and now you measure the functional connectivity in the model. And then you measure directly the relationship between the empirically measured functional connectivity with fMRI with the functional connectivity derived from the neurodynamic model, okay? And what you do, you just do basically noise, local noise, plus connectivity. At the group level, you explain about 0.4 correlation at the group level. At the individual subject level, you're explaining only 5%. And this 5% actually is really not explained when the subject is awake, while it is explained much better when the subject is in deep anesthesia or is asleep. And this is important. When you're in deep anesthesia, and this is work at Neurospin by Bartfeld, Dehaene, and others, when you're in anesthesia, your brain has a much less variable configuration that resembles the structural connectivity and is actually driven by intrinsic noise. But when your brain is awake, now the incredible multitude of patterns that you can generate does not relate it to the structural connectome. The structural connectome is not enough to explain the variability of behavioral states. So anatomical connectivity plus noise, which is one interpretation, holds only for anesthesia. Another idea derived from the hippocampus literature, where you know there are these place cells, Nobel Prize, Moser et al., and O'Keefe. Hippocampal cells, they show this interesting pattern of spontaneous activity after the mice runs the maze. And this post-play activity, what they call replay sequences, they show basically the same pattern of activity during the task, suggesting a role in offline consolidation. There is also, however, evidence from several people. There are also pre-play sequences. In other words, patterns of activity that are actually anticipating potential firing patterns during the mouse. Now, in humans, there is some evidence that the spontaneous activity that we see is actually related to offline consolidation. There is this nice neuron paper from the Washu group in which they cast an arm. This was Nico Dosenbach. They got his arm cast for 3 months, and they basically got scanned every week after casting his right arm and not being able to. He has accelerometer to measure the activity. And what he showed is that before the cast, there is good correlation in the motor cortex. During the cast, the correlation disappears. And as soon as the cast is off, the correlation appears again, immediately, within like minutes. And when you look at when this loss of correlation comes in, it's not immediate, but actually takes about 2 weeks to lose the correlation, which suggests that this loss of correlation may be due to learning of the fact that you're not moving your arm. So it's an offline process that is represented in spontaneous correlation. If it was an online process, then by wearing the arm and changing the dynamic, the biomechanics of your arm, then you should see that immediately. But the fact that you don't see it immediately suggests maybe it's offline, okay? Now, we have a theory we're testing, which is being described in this review in Trends in Cognitive Neuroscience with Marco Zorzi and Giovanni Pezzullo, who is a computational neuroscientist in Rome, in which we propose that the spontaneous activity is similar to a generative model in artificial intelligence, okay? So the basic idea is that spontaneous brain dynamics are manifestation of top-down dynamics of generative models, and we're going to try to explain that, detached from action perception cycles. Generative models constantly produce top-down dynamics, but under task condition, we call them expectation and attention. Of course, there is a lot of work on predictive coding and Bayesian inference by Friston and others, suggesting that the brain can, through these task control signals, anticipate the outcome of sensory signals and actions. But what we're proposing is that the priors of these inference models are actually coded in the spontaneous activity, are not task-driven patterns, but are spontaneous patterns. And so spontaneous brain dynamics during resting periods optimize the generative model for future interaction by maximizing the entropy of explanation in the absence of specific data and reducing model complexity. What does it mean? So, in cartoonish way, what we think is going on is that during development and through experience, your brain is exposed to a number of task activation patterns coming from different kinds of experiences that you have. Your brain, of course, has Hebbian learning and statistical learning, and through development and experience, these patterns, multivariate information states, not just connectivity and communication, but information states related to this behavior, will be coded in your patterns of spontaneous activity. And I think my mouse here is, okay, I'm going to use this. In the pattern of spontaneous activity. So at rest, what we're proposing is that the spontaneous activity is replaying these information patterns related to task states. And so when you're doing a task, now this pattern of spontaneous activity are going to be your prior for influencing the task during active behavior. They're providing the top-down biases. And there are, computationally speaking, two different goals of this activity, because during tasks, you want to emphasize the accuracy of prediction. So you try to minimize the variability of states to pick up what is important for that particular task. During spontaneous activity, what instead you're trying to maximize is the generalization of the possible explanation. So you're emphasizing the entropy of explanation, okay? So the idea is that your system under rest is developing these very complex general, okay, eigenpatterns. They're basically summarizing many possible behavior. When you're doing a task, now your brain is zooming in on a particular pattern and is using that pattern to basically perform a particular task. That's the idea. So there are experimental predictions of this idea, and we're testing this experimental prediction. The first experimental prediction is that we should see similarity between task and rest, not only in connectivity patterns, which is what has been done for the last 20 years, but actually in information state, multivariate state representing tasks within a region and across regions. And I'll try to explain what I mean by that. Secondly, these patterns should be shaped by learning and experience, and therefore we should see more intrinsic patterns for familiar and ecological stimuli than novel or synthetic stimuli. Because if your brain has accumulated this knowledge, then you're going to replay more likely patterns that are common and familiar rather than new pattern or synthetic pattern you've never seen before. And finally, and this is the third prediction, that the spontaneous activity pattern should not be representing examples of stimuli, individual examples, but they should be representing some low-dimensional state, some eigen representation of stimuli. Because during the resting state, you're trying to maximize the explanation and decreasing the entropy of specific prediction. All right, so we've done a couple of experiments, and we started in the visual system. Why the visual system? Because the visual system is well characterized, and there is also some evidence for low dimensionality in the visual system. So this is work by Nikos Christgordy at Cambridge, showing that when we look at human inferotemporal cortex, and you're presenting a bunch of stimuli, it turns out that the activity in human inferotemporal cortex can be classified, is highly correlated in terms of patterns. And you see basically two blocks. One has to do with animacy, stimuli, human faces, bodies, animals. And one has to do with inanimate, tool-made, human-made objects, scenes, buildings, tools, etc. So it looks like it's a low-dimensional structure in which it looks like that the activity in the visual cortex is divided between these two large classes of stimuli. Now, in monkey electrophysiology, there is also evidence that face perception is low-dimensional in the sense that by combining a limited number of dimensions, something in the order of 30 to 50 dimension, linear combination of spiking activity, I can represent thousands of faces. Okay, this is work by Dorit Tsao, showing that activity in the inferotemporal cortex can be represented by a linear combination of low-dimensional multi-population states. So we ask, okay, does human visual cortex replay stimuli before the stimuli were actually seen? Okay, and so there are two predictions. So that if I present, for instance, natural scene should be replayed in spontaneous activity more frequently than scrambled objects, okay, than synthetic stimuli. And the second prediction that, and this should be particularly stronger in areas that are object specific. For instance, in the parahippocampal place area, I should see more replay of scenes as compared replay of faces or other objects. The second prediction is that this activity should be represented averages, eigen canonical stimuli averages across many, many different individual exemplars, rather than individual stimuli. Okay, that's the assumption. So what we did, we basically got a paradigm in which we presented localizer, resting state activity before and after the task. We also localize category-specific region in human cortex at the individual subject level. And then we develop classifiers, stimulus classifier for different classes of stimuli, using something like representational similarity for those of you. So basically you're getting a fingerprint of different faces in different visual areas, a fingerprint of different scenes in different visual areas, and so forth. So first, the results, we get what everybody has seen since Nancy Ken Wisher in 1997. You basically get body-specific, category-specific region in visual cortex. EBA, extrastriate body area for body over objects, fusiform face area for face over objects, parahippocampal and retrosplenial cortex for scene over objects. Nothing unusual about that. Then we develop a method to compare these multivariate patterns in these regions with the resting state. So we got this template for individual stimuli or the average, and we slide those task templates, task patterns, we slide them frame by frame on the resting state activity in those regions, okay? So for each frame or resting state, you compute a spatial similarity between your task patterns and your multivariate resting state patterns. And you do it for 30 minutes of resting state. And now you develop a curve, which is this curve here, which is a curve of the correlation between your task patterns and your resting state patterns. And you see that the correlation goes up and down near zero. There are moments in which the correlation is 0.9. There are moments in which the correlation is minus 0.9. But most of the time, the correlation is zero. And now when you develop a distribution of correlation values, you have that distribution which is centered around zero and has a long tail of correlation, positive correlation and negative correlation. Is that clear? Okay, so I'm just measuring representational similarity in the resting state. So the hypothesis is that in the fusiform face area, we should have more frequently positive and negative matches because the fusiform face area is replaying faces. And this is exactly what we see. We see that if I build the distribution of stimuli, you see that it's more peaked at zero and is not as fat in the tails, while the distribution for faces is actually as longer tail, which suggests stronger positive and negative matches. If I go into the body preferred regions, and now I'm looking at template for bodies, I see the same thing. And if I go in the scene region, and I look at template for scenes, I see exactly the same phenomenon. So across all three regions, I see this more frequent occurrence or replaying of the target specific. So spontaneous activity replays in visual cortex are both object specific and area specific. Now, what about the issue of eigen representation versus individual exemplar? We have over 50 faces in the experiment, and so we can ask that question. And what we found is that the replaying frequency is much higher for the average template as compared to the single exemplars, okay? It's much more frequently replayed, the average, than the single exemplar face. So replays are more frequent for average than individual, so there is a low dimensionality to this replay, and this is true for faces, for objects, and for scenes. And then finally, which I think is particularly interesting, is that this phase replay, say face or scene or body, are not within a single area, but is a state that is emerging within the visual cortex. So if I take, for instance, multiple regions, they're representing scenes, two regions, they're representing scene, the retrosplenial cortex and the parahippocampal place area, okay? Or I represent two areas that represent in body, the EBA, the extrastriate body area, or the fusiform body area. And now I can do the same analysis and ask whether those two regions are showing correlated pattern of similarity when FFA is representing a face. Is the extrastriate, the occipital face area also representing a face? Okay, so it's a state of face representation. And this is exactly what we see. When we have a scene, when we have a peak in one area for scenes, at rest, we also see a peak for scenes in the other area. And you see there is a very good correlation which is not occurring in the body areas. And if I look at body templates, now I get very strong synchronized correlation in the body regions and no correlation in the scene regions. So what this means is that your brain across the whole visual cortex at that particular moment in time is representing faces or bodies or scenes. So category-specific replay in human visual cortex consisting of low-dimensional patterns occur across multiple visual areas. What about movement replays? Now, in animals, there has been some work for movement. I'm just going to do it quickly. We have several papers now that we're going to replay also actions in motor cortex. And what is particularly interesting is this is the experiment in which we're asking subjects to perform ecological movements like this or this. Okay, if you attach a sensor to a hand and you just let it run for 24 hours, with two components, you're explaining 90% of the variability. Those are called muscle synergies, in which basically you have a low-dimensional space of movement that representing many possible individual movement of your hand. So we'll look at movements that represent muscle synergy versus movements that are novel, like movement like this. Okay, so we compare ecological to novel, and we find that in the visual cortex, in the motor cortex, we see a much higher, stronger concurrence of replay in motor cortex for ecological scenes as compared to random novel stimuli. We also see this in association cortex. And finally, we see that dynamically, if I take an area, an ROI in the motor cortex, and I compute dynamic functional connectivity during these movements, in the resting state, I see exactly the same community replaying connected with the primary motor cortex. So even at rest, the whole brain is replaying ecological sequences of movements. Okay? So is this something, what I just told you, in the visual system and in the motor system of humans, is this something that is unique to humans? And so with Anastasia Di Marco, we have done an extensive analysis of the animal literature from C. elegans all the way to monkeys, and we found there is many, many examples of what I just told you. So I'm going to show you some limited examples. So for instance, in cat visual cortex, if you're doing optical imaging and you're running basically spontaneous activity through optical imaging, you get something that looks like evoke activity by orientation, okay? This has been shown during anesthesia. It's been more difficult to demonstrate it in the awake state. And when you look at the time course of the similarity between spontaneous activity and evoke activity, you see this peak of activity, which is exactly what we see. When you develop a distribution of correlation in cats, you see what we see in humans. You see longer tail in the primary visual cortex of cat, the primary visual cortex of monkeys, okay, and the category-specific regions in humans, our work. You go to the auditory system and you see exactly the same thing. Auditory replays in auditory cortex. So this is a general phenomenon that we see across species. What about movement? I told you about movement replay. There are many examples in the animal literature. I want to show you one particular example, which is C. elegans. C. elegans is a worm, and he moves, when he's moving, in a particular sequence. So, as is always the same sequence of movement, and you can now develop, record from all the neurons, embed all this activity in a low-dimensional space, and develop, extract statistics, trajectories, neural trajectories, okay? And this is what the C. elegans is doing. If you anesthetize the C. elegans, or you put the C. elegans in a gel, in which it's not moving, so it's not getting any information from the environment, it's not getting any somatosensory feedback because it's moving, the C. elegans spontaneous activity is going to replay exactly the same neural dynamics at rest, the neural dynamics that represent the movement. So again, this is an example of all low-dimensional representation of muscle synergies are represented in the C. elegans in the absence of any movement. So the key points of this part, and I'm done basically, cortical regions replay at rest stimulus and movement patterns, especially familiar and or ecological stimuli. Replay patterns compress information because we see an example of eigenfaces and muscle synergies rather than individual movement on individual faces. And finally, that these replay patterns circulate across cortical regions. We think through these spatiotemporal motifs that I show you in mice, those 50 patterns, okay, providing priors for perception and action. So there are many open questions that we're pursuing in experimental work. One question, I show you priors, replay in visual cortex, and I show you replay in motor cortex. But of course, we know that the brain is organized in hierarchies, okay? We have gradients. And so the question is how a visual prior gets transformed into a motor prior to a sensory motor transformation. So Esther Fusaro in the lab is going to look at how there is actually a transformation of these priors from visual cortex to motor cortex. Another question is, are these priors can be modulated by learning? And so Anastasia Di Marco, who's another graduate student, is actually doing sequence learning of visual areas, visual stimuli similar to the one that we've done in fMRI with high-density EEG, tracking replay sequences in real time. And I can tell you that we see these replay sequences, and like in the hippocampus, these replay sequences are shorter in time. A sequence that lasts 5 seconds is actually compressing time down to 50 to 100 milliseconds in cortical activity. And finally, the low dimensionality of behavior, which I think is a very important insight, because I think in psychology, we've been trying to split the cake of behavior too much, okay, in my view. Okay, that's one of the many virtues in psychology, but also some sins. And some of the sins is that we've been thinking about this brain as this modular architecture that with many different modules. And I think when you look at behavior, behavior is very low dimensional. So for instance, we've been doing experiments with eye movements in which we always compare a visual task with rest. And what we found that over many images and many observers, hundreds of observers and hundreds of images, with 3 components, we can describe all the eye movements of subjects. And it turns out that these subjects have different fingerprint of how they move. We have one group of subjects that have a longer fixation and they jump less. And we have other subjects that have shorter fixation and they jump more. And then we went back into the resting state activity in the EEG to see whether we can see trait of this behavior during tasks. And we have evidence that, for instance, static and dynamic differ in their baseline EEG features. And now we're using this information to study population like MCI and Alzheimer's disease, because we think that eye movement dynamics under natural condition is an excellent proxy for brain dynamics. And so by measuring eye movements, we might be able to anticipate what might be happening in a patient with Alzheimer's. So this is a healthy subject exploring a visual scene, an MCI patient, and an Alzheimer's patient. And you see there is an increasing rigidity in those patterns as people are exploring eye movements. So all this work is funded by many agencies that we thank here. Lots of students, most of them in red went to faculty position over the years. People in blue are people actually in the lab. The last thing that I want to tell you, just to leave you with a qualitative take-home message, is this. I told you about spontaneous activity, and I told you of these beautiful properties of spontaneous activity and cognition. There is one state that is particularly striking, in my view, about what the brain is doing at rest, which is dreaming, okay? Think about when you're dreaming, you're taking very high-level perceptual decisions. You're taking a very high-level anticipatory expectation. When you're falling off bed, you feel like you're falling off the bed. You're anticipating the fall. When you're flying, you feel almost vertigo. You meet people on trains and in time traveling, and you make moral judgments about people. Some people you like him in a dream. Some people you don't like him in a dream. And even when you're sitting in bed, you can recombine your past and your future. So the point that I'm trying to make is that even the highest level of human cognition, like empathy, in theory of mind, can be recapitulated by noise transition in your brain when your brain is completely disconnected from the environment, which really makes you think that cognitive activity is really not the way we've been thinking about, in which we're building this representation stimulus after stimulus or task activation after task activation, but a lot of the activity is prepackaged for you in these spontaneous activity patterns that you can reactivate during dreaming, also in the highest form of cognitive activity, the most human, like empathy and theory of mind. Thank you.